Hello and welcome to this video. In the next few minutes I will try to explain how a new technique is currently revolutionizing cancer research as we know it. This technique is called single-cell RNA sequencing, and it holds the potential to overcome so many problems we are currently facing in modern cancer research. Now, one of the key goals in cancer research is to identify the exact composition of a tumor in order to understand its biology and to uncover its weaknesses. This is crucial for finding a targeted treatment or even a cure for this specific tumor. Cancer was studied as a clonal disease for many decades. That means we thought that the tumor is made out of only one population of identical cells with identical characteristics. We now know that most cancers are not composed of only one population of identical cells, but rather of many different subpopulations, basically groups of cells that share distinct properties. We also know that those subpopulations can substantially differ from each other in terms of their properties, such as metabolism, motility, proliferation, invasiveness, metastatic potential, so-called stemness, and very importantly, resistance to treatment. This phenomenon is called tumor heterogeneity, and it is more and more appreciated in cancer research and treatment. But it is actually very difficult to decipher the different subpopulations within a tumor, and that is due to the way we usually process and analyze tumor samples. So in conventional cancer research, tumors are analyzed as a bulk. This basically means you take the whole tumor and lyse it. Now you have a pool of molecules like DNA, RNA, and proteins deriving from this tumor that you can analyze. You can then compare the tumor to healthy tissue and see the differences between the malignant and the normal pool of cells. But that's about it. So you might be able to see how the tumor as a whole differs from healthy tissue, but you don't know whether each cell in the tumor possesses the malignant properties you discovered, or if only a few cells within the tumor do. So instead of seeing how the tumor is actually composed, we see an average of all the cells we analyze combined. And as you can imagine, this average tumor does not depict reality very accurately. In fact, it hides all the different subpopulations and suggests that the tumor is made out of only one population, a population that combines all the features of the actual subpopulations, but does not really exist. Let me make an example to clarify what that means. So we know that cancer needs to acquire certain capabilities for tumor growth and progression. Those capabilities are also known as the hallmarks of cancer. When we look at the tumor as a whole, it seems to us that there is one clonal population that has accumulated all of those hallmarks. But it would be very difficult and time-consuming for every cell in the tumor to accumulate all those capabilities. A recent article in Nature Reviews Cancer suggests that instead of one clonal population accumulating all the hallmarks of cancer, the formation of subpopulations with distinct hallmarks is more likely. This could mean, for example, that one population is able to secrete factors that will help to form new blood vessels for the tumor, while another population can produce proteases that enable the tumor to invade its surroundings. This phenomenon is termed clonal synergy. The subpopulations within the tumor are working together and benefit each other. When present alone, the traits of each subpopulation are not enough to promote widespread malignant growth of the tumor. Only the combination of subpopulations is able to promote tumor growth and progression. This is of course very interesting and fundamentally influences the way we look at cancer. But the problem of identifying the subpopulations within the tumor remains. So we need to figure out a way to look at the individual cells within a tumor and find out the identity of those cells. But how do you even determine a cell identity? There are various ways you could attribute an identity to a cell. One very simplistic way would be to just look at different cells under the microscope and determine what they look like. This would be the equivalent of determining a person's identity by hair color. So that would give us a very rough distinction at best. Often we would see no differences between cells at all. What we want is the equivalent to a person's fingerprint. And we can actually look at a molecular fingerprint of a cell called the transcriptome. Now you may remember the central dogma of cell biology. which is that DNA is transcribed into RNA, which is then translated into protein. The transcriptome is defined as all the RNA molecules within a cell. So why RNA and not DNA or protein for a fingerprint, you might ask? Analyzing only the DNA cannot tell us which genes are actually on or off in a cell, and while the differences in DNA between different cells are often small, there are tremendous differences in transcriptomes. So why not protein? Protein would actually be very interesting to study, but there are good reasons to stick to the transcriptome. The proteome may not tell a cell's entire story. After all, proteins are dynamic and interacting molecules, and their changeability can make proteomic snapshots extremely difficult. Also, there are many technical challenges in characterizing proteins, because proteins cannot be easily amplified and have several post-translational modifications. But thankfully, measuring the intermediate step between genes and proteins, in other words, transcripts of RNA, bridges the gap between the genetic code and the functional molecules that run cells. So the transcriptome provides the best way to characterize a cell. So now we know how to uncover cell identities. But again, when we isolate RNA from a bulk sample, all the different cell identities are mixed, and instead of uncovering individual fingerprints of cells, we get one big smudgy fingerprint that can't tell us anything about the diverse cell identities within a tumor. So to look at molecular fingerprints, we need to look at individual cells. Now this is where single-cell RNA sequencing comes in. This revolutionary technology actually allows us to analyze the transcriptome of singular cells. But how does it work? So in order to get enough viable single cells from a tumor, the tumor has to be processed within a few hours after it has been removed from the patient. So a surgeon removes the tumor or part of the tumor from a patient. The sample is then brought to the lab and dissociated very carefully. This means the bonds between the cells are broken up while the cells themselves remain intact. The suspension of cells is then stained with two different markers in order to identify viable tumor cells. The first marker is called calcine and stains only viable cells. The second marker is called topro and it stains dying or dead cells. A machine is able to identify calcine-positive cells, meaning viable tumor cells. Those are the cells we want for analysis, because dying or dead cells already changed or degraded big parts of their transcriptome, making it impossible to uncover their identity. The machine can then sort the individual cells into a 96-well plate until every well on the plate contains exactly one viable tumor cell. This technique is called fluorescence-activated cell sorting, or FACS. The cells are immediately lysed after sorting, and the molecules of each cell are preserved in a buffer. After that, a complex cleanup process begins that is capable of isolating only the RNA of each individual cell. The concentration of RNA from a single cell is of course much lower than what you would get from a bulk sample. To amplify the transcriptome of each cell, the RNA in each well is reverse transcribed into cDNA. cDNA is basically a DNA molecule that has the same exact sequence as the RNA it derives from, so it carries the same information. But unlike RNA, cDNA is much more stable to work with and you can amplify it by PCR. The amplification makes it much easier to sequence the transcripts. So now each well on the plate contains the amplified transcriptome of one single tumor cell. The cDNA from each well is tagged with a barcode, a short, unique sequence of base pairs. The tagging allows us to pool all the transcriptomes together for sequencing. Thanks to the barcode, we can identify which piece of cDNA came from which cell later in the process. This way, the different fingerprints can be reconstructed in remarkable detail. The sequencing of the pooled sample takes about a day, and creates a ton of data. Processing this data usually takes a few weeks. What we basically get in the end is the reconstructed gene expression profile of each cell. This gives us a lot of information about the properties and characteristics of the cells, the fingerprint we've been talking about. With that, we are able to reconstruct the tumor architecture and get an incredibly detailed picture of the tumor's composition. The possibilities of data analysis are endless and include unraveling complex cell populations, reconstructing developmental trajectories, and modeling transcriptional dynamics. To discuss all the different analytic possibilities would be way beyond the scope of this video. So let me make one example that might begin to clarify how powerful single-cell RNA sequencing can be. By unraveling the different subpopulations within a tumor, we can identify the population that actually propagates tumor growth and progression. Knowing that tumor propagating population and its characteristics makes it a lot easier to find or design a very effective and targeted therapy to treat patients who are suffering from this specific tumor type. This targeted approach hits the tumor where it is most vulnerable. The tumor is also much less likely to develop resistances against a targeted therapy, leading to fewer or no recurrences. So single-cell RNA sequencing might provide an accelerated path to a cure for many cancers, especially those with complex compositions. A recent research project at Dana-Farber Cancer Institute and Massachusetts General Hospital in Boston utilizes single-cell RNA sequencing to identify the architecture of the deadliest childhood brain cancer called diffuse intrinsic pontine glioma, or DIPG. DIPG is a highly aggressive brain tumor affecting mostly children between 5 and 9 years of age. Currently, there is no cure and no effective treatment for DIPG. Most of the children suffering from this tumor die within 8 to 12 months after diagnosis. Single-cell RNA sequencing of DIPG samples holds the potential to finally come up with an effective treatment for children suffering from this devastating disease. To find out more about DIPG and this revolutionary project, click on the video on the left and visit fightdipg.org to find out what you can do to help this project. If you enjoyed watching this video and want to see more, please subscribe to our channel. Thank you for watching.